All right, so good morning, good afternoon, evening, wherever you happen to may be. I think we'll go ahead and start now as people kind of slowly start to filter in. So welcome everyone to the second virtual brunch, our sort of ongoing webinar series in this for these pandemic times. Hopefully we can maybe get back to some real conferences. Maybe ASMS will actually happen, but anyway, regardless, let's this is our kind of, it doesn't matter what time it is, you can join us for brunch and we'll talk about some science. We got a couple excellent guest speakers today. We also partnered with MS Bioworks out of Michigan for this webinar. We're going to talk mostly about DIA today, I think. I'm Jacob. Most of you know me from either talking on the phone or I've met probably most people here in person. I do sales and support for Proteome software. And I think we'll probably go ahead and jump right in. So I'm going to give it over to Michael Ford first from MS Bioworks. Like I said, we partnered with them for this webinar, and he's the first guest speaker up. So with that, I will stop sharing and take it away, Michael.
Good morning, everybody. Thank you for your time. Here we go. It wouldn't be an MS Bioworks presentation if I didn't at least plug the company somewhat. So just a little bit about us, what we do. We're a service organization based in the proteomics space. We've been doing this since, as MS Bioworks since 2010. And we've been working with Proteome software for a long time, and we've been using the Scaffold products for a long time. And we're pretty excited to see this Scaffold-DIA product come to market. I'm starting with an idea about what we've been doing and how it evolved and where we are now, and just going back a long, long time, relatively speaking, for some of you, with a look at the literature and how it's evolved and where we are now, from sequencing on triple quads through digesting with trypsin and present-day single-cell-based proteomics we're seeing come out, and distilling that down into a workflow that presumably a lot of people on the call will be familiar with and is really applicable to everything we do in the lab in one form or another, whether it involves fractionation or labeling or enrichment. These nodes are fundamental in the workflow. And if we look at how that workflow then is superimposed into the mass spec lab, and we look at the evolution of the hardware that supported what I'm calling an Orbitrap-centric workflow, we see that back in 2010 it was the Orbitrap Velos Pro, Orbitrap Velos Pro, with a linear ion trap and the D10 Orbi, and how that's moved through to present day, where now we're operating on in part on the Explorers 480, nanoscale chromatography, in terms of robustness, ease of use. We've seen a lot of technology come and go, but I think now we're at a place where we're happy doing UPLC coupled to these instruments. And then if we consider data-dependent proteomics specifically, we're looking again at the timeline of software and hardware evolution, quoting this from our perspective as a company, but then again...
Hey, Michael, sorry to interrupt. We're not seeing any slides. Are you sharing your screen?
Oh, yeah, hold on, sorry.
No problem. I just wanted to check if they were supposed to be seeing slides.
Oh,
right
okay.
now.
Sorry, I've got host disabled participant screen sharing.
Uh-oh, the host did something. That can't be me. All right, there we go. Sorry about that, everyone.
Okay, it's so nice. I'm going to show it twice. There you go. Okay, here we go. Is that good? We're good.
Yeah, it looks good now. Now we can see it. Thanks.
Cool. Cheers, Lindsay. Thanks. Okay, this is about the company, who we are, what we do, where we do it, and how long we've been doing it for. And it's pretty much industrial, academic, biotechnology clients, a lot of proteomics, and primarily data dependent. So this is the workflow I was describing that's evolved from the early days of some of the protein and peptide sequencing work to present day. And in one form or another, this is pretty much everything we do. And putting that in the context of the instrumentation, how the instrumentation's evolved, ease of use for nanoscale chromatography, robustness of the mass specs. Here we're going from essentially the Orbitrap Dollars Pro through the Q Exactive, Q Exactive HF, HFX, Fusion Lumos, and now Explorers 480. And they're all coupled with nanoscale chromatography in one form or another. And some of these are still in the lab, some of them have left the lab, but that's the way the field's moved from our perspective. On the software and hardware evolution side, hardware being computing power, obviously computing power has come down ridiculous amounts. So when we first got started, we were running Mascot from Matrix Science, Scaffold from Proteome software on the Beowulf cluster, and Dell workstations. And now we're running on PowerEdge servers with a lot more RAM and a lot more processing power, and really the cash investment there is not too crazy. But in this time, in this 10 years, we've seen some gains, and the gains have been primarily through advancements on the data processing side from MaxQuant, to a certain extent adopting one unique peptide reporting through it, second peptide identifications through MaxQuant, and then also match between runs. And those things combined have made label-free quantitation a little bit more attractive to customers. So with this increased sensitivity and robustness, that is less downtime on the instruments, the sensitivity gains have been pretty good. We've seen the high end, a 4-hour run, yielding something like 4,500 proteins. And this is with a QC we've been running for a very long time. It's a triptych digest from Promega. Promega helped MS Bioworks start up in 2010. And so you can order it from them. We inject 1 microgram, and we run this on the 2-hour run on all our instruments pretty regularly, every couple of weeks or so, independent of daily QCs, where that's just a BSA triptych digest. But really what you're seeing here is gains, incremental gains across a 10-year period. So in the background, as a service company, you know, we've been approached by a lot of people doing different types of things with data-independent acquisition in one form or another. And we knew that the folks at Proteome Software had a product, and that was evolving. And we were watching the space. And so the cartoons here essentially show the difference between the 2 acquisition methods, where in one, we're isolating a precursor, fragmenting it, and in the second, we're isolating a much bigger window and fragmenting everything. And so there is a computational challenge there that's been addressed by a bunch of folks, and there's some really cool stuff in the literature. And the paper I cite there is one that I found to be a really good read to understand the space. So we were looking out for opportunities to get into the space, and in 2018, we got the email from the ABRF. They do these kind of inter-lab studies that are really useful. And this one was for DIA. So we thought, okay, we'll get in here because they've got expert panelists and so on and so forth. And what we were looking for here is methods and how we were going to run things internally. So we're lab number 29. There's a paper associated with study that's on bioRxiv. You can get a bunch of information from there. But we were quite happy with the outcome, and we were impressed by the sensitivity. Just over 4,000 protein groups in 2 hours. So we thought, okay, that's pretty good. So we decided to explore it further. And then in 2020, when we had the lockdown, we were talking to the folks at Scaffold, and they said that they were really pushing the product now, that they were going to do a lot of development. Because this this figure's from the manual. The Scaffold-DIA manual, by the way, is absolutely phenomenal piece of work. It alone is worth reading. But this is one of the figures, and it's a pipeline for processing DIA data, and it's a little bit overwhelming. It can be overwhelming, and if you were to try and piece this pipeline together in your own lab from all your academic resources, it's not turnkey for sure. I'm sure in the right hands it would be, but if you're coming at this from a perspective of a greenhorn, it's not necessarily turnkey. So the DIA product then has essentially brought together all of these parts, absolutely amazing pieces of the puzzle, and put it all into one product, which runs on Windows. It's ridiculous. And everything that's here, I've put in the citations I could find, and I'm still we're still getting into the space, and so it's fun to be reading this stuff. But some of these products, some of these technologies are absolutely phenomenal. The Prozit, if you haven't sort of played around with it or even read up on that, worth a look, just phenomenal. The encyclopedia paper, and then incorporating percolator and having it all available as one package is really phenomenal, really phenomenal. So what we were particularly excited about was this development, and this was the idea of spectrum library-free approach to DIA, where we can use gas phase fractionation to make a library. And this is very attractive because from the experiments we've done, we've seen fantastic sensitivity with minimal overhead and really no concerns related to library generation. And again, this pipeline is made possible by Scaffold-DIA in our lab. And I put a reference down there. Absolutely go read any references you see here. I've thoroughly enjoyed reading. So we're going back now to our sort of evolved pipeline. And we're taking a method from the literature and bringing it into our lab and reproducing it with no other changes beyond instrument settings and data processing. So we were very excited to do this. Excalibur now running on Windows 10. It has data acquisition options in there that can configure with reference to the literature. It's very easy to bring this on board. So this is kind of the punchline with reference to slides I'd shown earlier about incremental increases in sensitivity. This is as significant a gain in sensitivity we've seen in 10 years with millions of dollars invested in hardware. So that's a 30-minute run, and it's over 5,000 proteins identified in a 30-minute run. And so we've seen some stuff in the literature about biofluids, applicability there. That all looks very interesting, but just screening cell lines alone, this is a massive opportunity. So in this particular example, then we wanted to show the software and the data acquisition method. We wanted to kind of put it in a position where we could really highlight the sensitivity and what it can be used for. So in this particular case, we have samples from gene-edited cells. And when people do gene editing, part of that pipeline that's highlighted here is selection of clones. So it's single clone culture and then some sort of assay to determine which cells have been edited. And a Western blot is a popular choice for that. And so obviously, depending on where you're sitting, Western blot is kind of like it's up for discussion, right? But if you can do targeted PRM, that's great. But yeah, the single cell culture and then some sort of quantitative assay to help guide cell selection. So we had 34 samples with 90 minutes per sample, and across the 34 samples, there was 6,100 proteins that are common. This is label-free quant. Absolutely, in the context of the workflow I showed, there was nothing here that needed doing to the samples after digestion. So digestion, solid phase extraction, onto the mass spec, and then 90 minutes and 6,100 proteins common, and a total of almost 6,500 proteins in 90-minute runs. And then this figure can be used to view which are the edited cells, which are the unedited cells, and then also to identify the edited proteins. And if you take because you've got so much data, right, you can do some pretty nice tests. And in this particular case, then, we've got this t-test followed by hierarchical clustering analysis. And this is so much more depth than you would get from a Western. Absolutely ridiculous. And it's a 90-minute run per sample. So we're redoing this whole study now with much shorter runs to basically package into a one-day experiment on a similar number of samples that can be used to guide some pretty significant decision-making, really. And this is just one example. So, you know, from the perspective of MS Bioworks and our involvement with Scaffold, Proteome software, and now having Scaffold-DIA online here, what do we see? We see quadrupled budgets for proteomic profiling. That's essentially the message. That before, where we would say we're going to do this experiment with a 4-hour profile, now we're going to use a 1-hour profile. And going back to 2010, where a deep profile experiment would be a 24-hour experiment on the Orbivelos Pro, to present time, where we're doing a lot of proteomes with 4 hours of instrument time, with UPLC using data-dependent acquisition on the Fusion Lumos, one of the Fusion Lumoses in the lab, or we just have the one Explorers 480 or on it. And no matter what size group you're talking to, people always want to do more experiments, and budget is always an issue. And so to offer people the opportunity to do 4 times as many experiments without changing the budget is huge. So, and we wouldn't have brought this online here if it wasn't for this product. So we are super excited about this product, super excited to have it in the lab, and really looking forward to bringing this workflow to market, really, and seeing where it goes. But it's a massive, massive advance, and we're appreciative of the work that the folks at Proteome Software have done here. And with that, I'll hand back over to Jacob.
All right. Michael, thank you for that presentation. We are obviously, of course, very excited about Scaffold-DIA as well, and we're pleased and excited to see that you have found the results so beneficial and are looking forward to seeing some more stuff that you all are doing over there with DIA and getting some more feedback and everything. That's awesome. All right. I'm going to give just a brief presentation. I have a couple slides here. Oh, I forgot to mention, if anybody, I should have done this at the beginning, I apologize. If anybody has a question, feel free to post it in the Q&A. We're monitoring those, and we'll try and get to those as we go. And if not, we'll save them for the end, and we'll follow up with everybody who has a question. We'll try and get them all answered, if not during the actual webinar, then afterwards. So I just have a couple slides here, some updates from Proteome software. We have been hard at work over the past couple months. Hard at work, I think, is kind of an understatement with all that we decided to take on at the same time. But it's all here. It's finally done. We late last week released all new versions of all of the software. So you'll see the new product logos up there. We updated to Scaffold 5. And for the first time, I believe in the Scaffold line, in Scaffold itself, it reads raw data directly. It does this through an interface with MS Fragger, which is a very popular search engine right now. This is something that people have been, I think, wanting Scaffold to be able to do for a while, is interface with MS Fragger. A separate MS Fragger license is required, so you'll need to, which is free for academics, so you'll need to go in and download that and then connect the two programs together. But it's pretty cool to see Scaffold read raw data for the first time ever. That's one of the big new features. There's a ton of big new features and bug fixes all across the lineup. You'll see Scaffold Perspectives, you might notice, is missing from that product lineup. We've replaced that with Scaffold LFQ. It's will work pretty much the same way. It should be familiar and intuitive for everyone who's used to Scaffold to be able to pick up on that. That's part of the sort of program changes that we've been making over the time. And another big thing that we did during this that's related to the software is we've converted everything to the new, more flexible key system that people who already had Scaffold-DIA or Scaffold Perspectives will be familiar with. Gives us way more flexibility, much easier to manage there. So for everybody who has an existing or an active support license, you should have gotten a license key already for the new programs. If not, feel free to reach out to us and we'll make sure to get that taken care of. In conjunction with this, we've released a new website also. The website hadn't been updated in a little while, so it's got a exciting new look. It kind of matches everything on this kind of big branding strategy that we've been thinking about. It connects directly, as it did, to our help center, which has tons of great resources. If you have any questions about using any of our programs, there's user guides up there. There's some helpful demo data. There's frequently asked questions. There's all kinds of stuff. Great resources for those getting started with DIA, since we've been talking about that a lot, for creating libraries and things like that. There's just a lot of good features up there. So go and check out the new website. You can download all the new programs are up there. So everybody that has a active license can go get the new versions. Like I said, we should have sent new keys out already. So that's kind of the other big thing that's come around. It's all new here at Proteome Software. And finally, if there's any questions or comments that we don't get to today, feel free to reach out to us. You can contact us at support@proteomesoftware.com, or you can go through the website to open a support ticket with us too. So I think that's all that I have. Like I said, I just wanted to talk about our new lineup briefly. I am now going to pass it over to Sue Weintraub from the University of Texas Health Science Center in San Antonio. And she is also going to talk about DIA. So I'm excited to pass it over to her. So let me do that. And Sue, I will pass it to you.
I'm getting there, just finding the right. Okay. Thank you very much, Jacob. I'm delighted to be here and presenting to this group. As you can see from the title of my slide, I think DIA is absolutely astounding. And I consider it the most exciting technology I've used at least the past 20 years. We've been doing DIA analysis for at least 2 years, 2.5 maybe. Really waited until the Scaffold product was available because I wanted a clean and easy workflow that could be used in the context of a core laboratory. I'm showing here our general process method. This experiment was now done a few years ago, closer to the beginning when we optimized our scan settings. It was 4 experimental groups of 3 replicates, just showing using S-traps for sample prep, the MS analysis of 2-hour gradients, the Scaffold-DIA processing. Now, it's a day, but we do, as the samples are acquired, convert them as needed to the .dia format. But the total time was only 5¼ days, and we got 6,000 proteins. Of note, here we were using 6 gas phase fractions to make the chromatogram library. We're now using 3. Fairly early in our use of, or really exploration of DIA, we had this project come in from Alex Bishop, where they wanted to look at differences in the chromatin fraction of cancer cells after the cells had been manipulated in a certain way. We have traditionally done most of our mass spectrometry and proteomics using gel-based, what we would call gel CMS. And it really is very nice to be able to look at a sample on a gel to get a sense of the complexity and relative quantities. In this case, we didn't have a protein concentration at this point, but loaded different volumes and could see quite a few proteins and fairly evenly distributed. Then using our standard process, gel is only run about 1.5 centimeters, cut the gel lane into 6 slices. Each slice gets reduced and alkylated, digested with trypsin, analyzed by MS this was for DDA analysis. Then we use Mascot for database searching, and then assembly into Scaffold. These were run on Scaffold 4. Now the new version is Scaffold 5. And you see the layout that I'm sure you're familiar with. And I'm just calling it cell 1 and cell 2 because this work isn't published yet, showing in regular Scaffold 4 samples view the total spectrum counts. You can do a t-test as shown here. And the green is representing statistically significant with multiple testing correction. You can notice in the pink area, there were 1,766 proteins identified. If I sort by smallest to largest p-value, you can clearly see that there's some striking differences between the experimental groups. Scrolling all the way down, there were 788 proteins that were significant with equal or less than P022. That's a fairly large proportion. So we really had already given the investigators some interesting information, but decided this would be a perfect test for DIA. And here is the samples view in Scaffold-DIA. And what we always look at first is the run order. Our samples are blocked and randomized to minimize any kind of systematic error. And it's always very comforting to look at the run order and not see a pattern, for example, a constant decrease in quantity or increase or something that would indicate an instrument or processing problem. The next thing we usually do is the principal component analysis, because I can't wait to see, are there going to be some interesting differences? And in this case, not surprising that there's very striking separation between the 2 cell types. And there's even 2 dots right on top of each other here. Now, you'll notice that I've highlighted exclusive intensity, and this is an absolutely key point. It's also fairly important looking at non-shared peptides in spectral counting, but here it's really critical. In the similarities view, or similar proteins, which I really love having in DIA, and you see that also in the LFQ, what you notice for PLRG1 is there's quite a few peptides that were identified and quantified. Nothing is shared, so you don't have to worry about this protein. But for PPB, and that's a serine-threonine protein phosphatase, there's quite a bit that is shared. And what you see is there are three forms. These are the catalytic subunits of B, A, and G. And A has the most that is exclusive to this protein. As you will see, some places call this unique, but really exclusive is the most appropriate name because it's exclusively assigned to this protein, while B and G have less proportionally. Well, why does this matter? Look at the exclusive intensities. These are the summed intensities for all of the peptides assigned to the protein. And you see that there's a very large difference between the quantity of A and B and G, and then a different proportion in the two cells. If you counted all of these peptides, say for isoform G, you would get a noted overrepresentation or overemphasis. The quantity would not be correct because you have something that's much more abundant that's being counted. So it's really important to use for all of the quantitative data only the exclusive peptides. Now, also something that I love, it's been an important feature of all of the Scaffold products, is I like to look at data, and I'm not happy just getting a report of an Excel spreadsheet where I can't examine what's happening in the spectra. In DIA, you're not going to have a standard spectra that you're used to from DDA, but what you can see is the peptide fragments that are co-eluting, and these are the ones that are being used for quantitative analysis. The ones that are colored are the ones that are counted, but the others you can see are also co-eluting. The precursor is shown here, but it's not used for quantitative analysis. But what this is important to think of is, picture a DDA spectrum where you have a pattern, and you expect that that pattern is going to be the same for other samples that have that same protein and that same peptide. The proportion of those peaks should be the same. And that's what you can see in the fragment intensities view, where even though the absolute quantity of each of these is different, the relative proportion is the same. And this gives us confidence in the quantification and identification. Now, you don't do this obviously for every one of thousands of proteins, but when you have a protein that's of particular interest, and especially if it's low abundance, you want to be able to examine it yourself and be sure you have confidence in it. All right, now back to the results. Again, going to do a t-test and sort by p-value in the same way. Green here is significant with correction for Benjamini-Hochberg multiple testing. And you really can see the notable difference in the relative quantities of this group. I'm going to scroll down again like I did for the spectral counts. Now there's 2,465 that are significant. Notice here, the ones in gold, those are less than P05, but do not pass the cutoff for Benjamini-Hochberg multiple testing correction. Then I wanted to see, here's it showing in the log2 screen so that you can really get a clear indication of what major difference there are in these two cells. But I wanted to see what was the comparison with the DDA data. And this is the set that I had just shown you. Going to highlight two specific proteins, DYHC1 and TCP4. And shown here is the corresponding DIA data. These are the same lysates. Obviously, they're different samples because the DDA was from a gel CMS and the DIA was from a single S-trap digestion and sample prep. And what's really notable here is we had two spectral counts in each of these cell twos. You do see a good, reproducible, and fairly similar intensities. But here we had 0. And does that mean really nothing? No, it's just a lower relative abundance, and it did not trigger any fragmentation spectra. Likewise for TCP4, here we have, you have a higher number of spectral counts, and the absolute abundance is higher, not absolute abundance, but the summed intensities from the DIA peaks. But over here you had 2 and then none. But in fact, it just meant that fragmentation wasn't triggered here. And if I needed any convincing of how great DIA was, this sample set made it very clear, and to the investigator. And there it is in the log2 form, where we're looking at the relative to cell 1. Now, this was a very interesting set that we ran by DIA. And here I'm not showing you the run order, but it was a set that was from male and female mouse neural stem cells. And the experimental design was there were 3 mother mice, and then from each mother there was a male and a female that was selected. The neurostem cells were acquired and put into 3 separate batches for each one. And then it received either vehicle, dose 1, or dose 2 across the 3 sets. This is the PCA for that group. And you'll notice a really amazing clustering, because it turns out that it clusters by mother and by sex of the mouse. And what this shows you is that if you hadn't looked at them separately and you just pooled together by sex, by all the mothers as a separate experimental group, you're not going to see any statistical differences. But instead, if you do a multi-level statistical test within a mother and within sex, then you get to start seeing what the true differences are relative to the doses of the drug. So this is showing an amazing aspect of being able to do the PCA and associate the changes with the different groups. Now, in this set was some prostate cancer cells that the investigator had done some knockdowns on. And first, again, I'm looking at the run order. And in the log2 fold change view, you can really see the patterns well. And there doesn't seem to be any problem related to run order at all. Now into the experimental groups. This is the wild type. Nothing is done, no treatment. This was a sham treatment. This group is a knockdown of a particular protein, one set and another. And it was pretty disturbing because it didn't seem to fit into the experimental groups the way the investigator thought. Doing a PCA, what we could see is they really didn't seem to fall well. Now, this is the completely untreated, and this group is the sham, so that's not too bad. But these others really don't behave the way we thought they should. And seeing here, this was set 1, so this is designated 51, 1, and 2, and then here's the 49s. And I said to the investigator, have you done Western blots on this? How did you verify about your knockdown or knockout? And in fact, here they are. And 59, 1, and 2 were really knocked out, but 59, 3 wasn't, and the other 49s. And so I'm not sure what was happening at this point. And then she started thinking, she says, oh, well, each one was a separate clone. And it surprised me that she hadn't done a Western blot on each one first before submitting the samples, but she's gotten some very interesting information on differential levels of knockdown. But the mass spectrometer doesn't lie, so we could tell that something wasn't the way she expected. And here's some fairly recent data that's very exciting to me. This is in a urothelial carcinoma cells, bladder cancer, where the cells are isolated from individual tumors from separate subjects. Again, looking at the run order, making sure that there's no problem. Looks fine to me. There were 4,816 targets. Now these were cells from, again, each one is a separate subject. the investigator thought that there was going to be a difference based on the grade of the tumor, but there was no apparent correlation between tumor grade and relative protein quantity. When we looked at the PCA, you can see kind of interesting pattern. So I arbitrarily picked L, which is left for this group, the one that's in the middle here, and the ones on the right, just to see what would happen. And if you do a t-test, and this is comparing L versus R, what you see is a large number of statistically significant differences with Benjamini-Hochberg multiple testing correction. And this was quite amazing. So immediately, and I will say that Brian Searle directed me to not jump to conclusions that these were biologically relevant. So we went back and had the investigator look at all the different aspects of sample acquisition, the tumors, how they were prepared. And really, there were no differences that they could find that were in any way related to sample prep. If you take this data set, then you can very easily export into Excel. And excuse me, I jumped ahead, but I wanted to show you this volcano plot of the data, which strongly shows a large number of significant differences. And if we pick this spot and look at the results, you see it's very convincing that there's something quite different about this protein and many others in the R compared to the L. Exported the results into Excel and then divided into the proteins that exhibited a 1.5-fold higher level of change versus up versus down, and submitted this to Reactome analysis. Reactome, if you haven't used it, is a fabulous site out of the UK where you have no charge and you can do pathway analysis, and it's especially powerful for human and mouse and rat also, but especially for human results. This is the foam, React foam image, where the size of a block corresponds to the number of reactions of proteins inside. And the lighter the yellow, the more of your proteins that you submitted that are overrepresented. And we can see here that there are some very clear pathways that are overrepresented by these tumor cells. That's the ones that are 1.5-fold down, and in 1.5-fold up, there's different reactions. I've often found that this is, there's a difference between up and down that's worth looking at them separately. Now, this is something that we do for all the investigators who submit samples. I'm happy to work with them and do the interpretation because many have no experience in proteomics at all. And it's part of what I feel is essential from a core to be a collaborator and not make it as a service. Now, this investigator is intrigued by the results. We're planning now to analyze a very large number of samples so that there can be enough population in the different characteristics so that we can find statistical significance or biological relevance that related to an attribute we can understand. I have to thank Sam Pardo and Dana Moeller. They have done always a great job, but especially during the pandemic, they have been working split shift and keeping it productive, and they're just extraordinary in their work. I will also thank the Health Science Center and the Mays Cancer Center, NIH and University of Texas System. I perhaps didn't mention again, those were all run on Illumos, and the UT system paid for that Lumos and Proteome software, and it's been wonderful working with them. So thank you, and I'll be happy to answer any questions now, or anyone who would like to look at data with me in the future, we can set up a Zoom session and I'll be glad to show you and or look at yours. Thanks.
Excellent, Sue. Thank you very much for
I realized I didn't have my video on, but that's okay.
Thank you very much for giving that presentation, both on looking at some interesting data and using Scaffold-DIA to analyze that data, and using Scaffold-DIA to kind of figure out that something was wrong, and then reaching back out to the investigator and having them do a little bit more digging. And it's cool that not only were you able to use the program to get some results, but you were able to use it to do some kind of troubleshooting there. So we like to see stuff like that.
Right. I can tell you there's a data set running right now remotely on the lab, and I hope it'll be finished by the time we're finished and I can get to see some even new data.
Awesome. Cool. Yeah. Cool. All right. Well, I think it's time for our last guest, certainly not least. I'm going to pass it over to Lindsay Pino right now from the University of Pennsylvania. So we can stop looking at my boring schedule slide and start looking at some actual DIA science. So go ahead and take it away, Lindsay.
Cool. So just to check, are you seeing my slide screen or are you seeing my presenter view?
Slide screen looks fine.
Perfect. Every now and then Zoom switches it up on me and it goes backwards. So I really enjoyed the two talks today, kind of going over both the current state of what we're doing with data independent acquisition. And I wanted to launch off of that, maybe a discussion of some of the future things that we can think about doing with DIA quantification. And I'm going to present that in the context of doing these kind of dynamic spatiotemporal proteomics. So just really quick, I do have a couple disclosures. I'm currently employed as a postdoc researcher at the University of Pennsylvania, but I have also co-founded and I currently consult for a company called Talis Bioscience. So in this talk, as I mentioned, we're going to focus on some of these maybe future directions that we can think about DIA quantifications pushing the field of mass spectrometry proteomics. So I'm going to start off introducing a concept that I first proposed a couple of years ago based on doing quantifications in the context of a reference material. Then we're going to talk a little bit about some work on DIA for exploring protein turnover. I'll talk a little bit then about PTMs. I saw Christina's already got a couple of questions, so hopefully we can make that a bit more of a discussion rather than a lecture. And I want to mention there's other aspects of spatiotemporal proteomics that don't currently have a lot of DIA applications, but I think could definitely be futures for DIA work. So let's start talking about this reference material because it's going to kind of be the foundation for the rest of the talk. So I'll start off just saying, and I think Sue has really just demonstrated that really well, that mass spectrometry itself is not inherently quantitative. So as mass spectrometry proteomicists, we know that you can take a sample, and even if you have the exact same sample, biologically, replicates can have some differences, or even technically, replicates can have differences. And that might be because we have two different operators. If I have two technicians or two postdocs, or maybe even two PIs, prep the exact same sample, they think the exact same way, there's going to inherently be variation between those two samples. And then, of course, as we know, especially in core environments or in labs where you might have multiple of the exact same instrument, a dirty instrument versus a clean instrument, or the favorite instrument versus the one that maybe nobody ever signs up for because it's a little bit lower signal. All of these aspects can cause two samples to have very different perceived quantities when actually, truly, biologically, they had the same quantity. So what we're going to do in quantitative mass spectrometry proteomics is we're going to compare the two chromatograms, and we're going to say, oh, this chromatogram has twice the intensity as this other chromatogram, as Sue has shown us now. And ideally, that would be the case. Ideally, if I had one sample and I measured a particular signal, I hope that that signal is related to the quantity of the peptide that was present in the sample. If I measured a second sample and the signal was 3 times lower, I would hope that that quantity is also 3 times lower, but we know that's not the case. For example, we might have two instruments. So if I measured one sample that had truly a high quantity and measured it on maybe my dirty instrument, or measured it on my instrument that doesn't perform quite as well as my workhorse favorite instrument, the signal of that sample could appear lower than the signal of a lower quantity sample that was acquired on my favorite instrument. And this is because those two signals off of those two instruments is not calibrated. So if we look at one chromatogram versus a second chromatogram, even though the bottom chromatogram comes hypothetically from a sample with more peptide quantity in it, because those two signals aren't calibrated, if I were to just compare their raw intensities, I would think that the chromatogram on the top corresponds to a higher quantity than the chromatogram on the bottom. So in quantitative proteomics, you have a lot of ways of trying to get around this. Most commonly, people try to use internal standards where you order synthetic peptides, maybe winged synthetic peptides, or maybe if your lab has a lot of money, you can get into synthetic or purified proteins. You spike those into the sample, and then you can compare the endogenous peptide to the labeled peptide and use that quantitative ratio for your studies. But we're doing DIA, that's absolutely not going to be possible if you're trying to measure every single peptide in a sample. You can't get synthetic peptides for everything. So instead, what we have been doing is using external standards. So you would have your experimental sample of interest, and then you would choose some kind of reference material and then compare those two chromatograms to each other in order to make a quantitative difference call. So what would your reference material be? It really is whatever your experiment happens to be. So if you're working in cell culture, you would pick a particular yeast or bacteria or cell line to work as your reference. If you're in biosamples, you might need to purchase pooled plasma or pooled CSF. But alternatively, you could also just pick a diverged species. So if I'm working in yeast, maybe a diverged enough species could be bacteria, where the peptides in the reference are different from the peptides that are in the experimental samples. Whatever it is, you just pick some reference, and that's what you're going to be reporting all of your quantifications relative to. So that's kind of an introduction to how we might approach quantification in some of these spaces where DIA doesn't let you get stable isotope labeled standards. We don't need those stable isotope standards to perform good quantification. So other ways people do quantification, especially with DDA, is using SILAC, stable isotope labeling with amino acids in cell culture. So you would take sample A, you would take sample B. Thanks for the laser pointer assist. You would take sample A, you would grow it in light media. You take sample B and grow it in heavy media. When you mix those two together, you could then compare internally within a single mass spectrometry run the relative intensities of the light to the heavy peptides. And of course, with DIA, we have more of this reference material approach where you'd have the two separate samples, but then you might have more variation between instrument to instrument runs. So it becomes a very natural question, well, why don't you combine them? Do SILAC-DIA. So there's some reasons that maybe you would not want to do this regarding splitting the MS1 isotope precursor peaks across different DIA windows. But what we did is we decided to just ask the question empirically, can you combine these? So we took a light proteome and we took a heavy proteome, and we created different ratios of these two mixed together. So a 100% heavy sample, 10%, 1%, 0.1% heavy sample. We're going to compare the quantifications between the light and the heavy ratios. So this is showing that data. So we have those expected ratios shown on the vertical axis, and then on the horizontal axis, I'm just spreading out the different ratios by the log2 abundance of the heavy peptide. So when we look at these ratios, we would expect a measured ratio to fall along this solid horizontal line. And that's more or less what we see. We're measuring this with DDA, and we see, especially at these easier to measure ratios, such as the 70%, 50%, and 10%, that DDA is pretty accurately capturing the correct ratio in these samples. We see that ratio measurements start to decay a little bit once we get down to these more difficult to measure, more extreme ratios like the 1 and the 0.1% samples. When we measure the exact same samples by DIA, we see that's not the case at all. So we get an extra order of magnitude in additional, more sensitive coverage of the extreme 1% ratio. So that's what I'm showing in these red boxes. We get an additional 1 order of magnitude sensitivity in ratios, but we also get an increased dynamic range. And that's what I'm showing along the horizontal with that arrow. The dynamic range with DIA is just a lot broader, because with DDA, you're only picking those top 10 most abundant. And even if you're using match between runs, you're not as capable of digging quite as deep into those lower sensitivity, lower abundance peptides. So clearly it looks like with DIA, you can dig a lot deeper into these ratios that you typically see with a protein turnover experiment, with a pulse SILAC experiment. So we're sold on protein turnover being improved by DIA, and I wanted to talk a little bit next about how DIA might improve post-translational modifications. So this is some work by another postdoc here in the UPENN lab, Josh Baeza. What he's done is he's taken all of the PTM modification sites that have been detected over time. So clearly for a long time, it was very difficult to find new PTM sites on proteins. And then there's suddenly around 2010, a huge jump in the number of acetylation sites that we knew about. And this is because now mass spectrometry is being applied to PTM discovery. And that's not just acetylation. What Josh shows is it's the same for phosphorylation too. Once mass spectrometry is introduced into the field of PTM discovery, we get drastically more modification sites discovered. So just kind of overlooking the field, there's a lot of different PTMs that we can imagine wanting to quantify. Of course, phospho is one of the top favorites, but things like acetylation, glycosylation is becoming very popular to study. Simulation, ubiquitination are all extremely popular PTMs to be studying these days. And in part, these increased discoveries are because of improvements in the sample preparation and just looking at different types of biological material and perturbations. But when I look at the top end of this figure that Josh has made, I see it kind of starting to plateau. And part of me wonders, is that a true biological limit of the number of PTMs that exist? Or are we just kind of hitting the limit of what we can do with conventional measurement methods that tend to be DDA based? Maybe we could improve this plateau by using DIA based approaches. So one of the things that gets a little tricky when you're going to try to do DIA with PTMs is the idea of isobaric PTM. So a peptide modification where that modification might be on different residues within the same peptide. So this is something we think about a lot in my postdoc lab because we study histones. So super, super modified, lots of isobaric peptide species. And what I'm showing you here is all of the DIA precursor isolation windows on the vertical, and then those DIA windows separated by the average measured retention time. So I'm showing you just one slice, one cycle of a DIA isolation window scheme. And over time, at this one particular retention time that I've picked, we can see that in these windows, for example, 3 precursors are observed in the 50 MZ window from 750 to 800 MZ. We see 1 precursor in 550 to 600 MZ, 3 precursors in the next window down, and so forth. So when I expand that out and show you the full cycle, looking for histone PTMs in this particular DIA method, one thing that you see is that there are a lot of DIA windows, isolation windows, where we're co-fragmenting lots of isobaric species or species within that 50 MZ range. This is not super ideal, right? An ideal DIA method, we want as few co-fragmented isomers as possible. We want a fast cycle time to get points across the peak. And we don't really want to waste instrument time on idle windows. So we don't want to have really high ion injection times trying to collect precursors that aren't there in the first place. So when we're thinking about optimizing this kind of method, one of the approaches that we advocate for is the idea of staggered windows. So then you get half the window size for the same cycle time on your instrument. And another thing that we can think of is decreasing the precursor range that we're measuring. So one thing, if we decrease the precursor range, now we have less time that we have to spend acquiring data. If we decrease the window width, now we have more specific windows. We have smaller window size, so we get more specific sampling of the MS1. And now when we start to look at some of these windows, we can see that with the smaller windows, we've split some of these co-fragmenting isomers into 2 separate windows. So now I have, instead of 3 different precursors being co-fragmented together, now I get 1 and 2 precursors in 2 separate windows. You can see this happen a couple times throughout this. Now I'm highlighting another where we have 2 precursors that we're able to separate into 2 different DIA windows. Now we're no longer co-fragmenting those precursors. Oh, and this is just to say that this is just one particular DIA method optimization for one specific assay, but you can easily imagine these kind of principles being applied to any other type of DIA that you're doing, because it really all depends on the sample preparation. So for example, Christina doing immunopeptidomics, when you're looking at some of those things, a lot of the peptide size might only be 8 to 9 amino acids long if you've got a good purification. So that might shift the kind of DIA scheme that you want to set up, because now you're looking for peptides within a more constrained mass to charge, precursor mass to charge range. So there's some established DIA MS workflows for PTMs, especially the work that Brian Searle has done in the past few years. Typically, these start with acquiring a DDA spectral library, and then you can use that DDA spectral library to search your DIA data. And then there's, like these great algorithms for localizing site-specific PTM fragments to ensure that when you have these positional isomers, that you're quantifying just the correct one. But I wonder in the future, we can't do this right now, but in the future, can we replace that DDA spectral library, instead use predicted PTM spectra? So this is something Dr. Ford had mentioned earlier, using algorithms like PROCIT. So right now, currently, I don't think PROCIT can support PTMs like phosphorylation or acetylation, but you can easily imagine this being future work, like near future work, that we no longer have to acquire these DDA spectral libraries, which requires just so much material and extra instrument time to get deeply fractionated spectral libraries. So a very quick overview of some of the spaces in which DIA is starting to be applied, and I think could be applied in the future. But as summary points, DIA and mass spectrometry is not inherently quantitative, but we can perform really high quality quantification without getting stable isotope labels like other targeted approaches do. Using DIA increases accuracy. So if you're doing things like protein turnover with a pulse SILAC-DIA, it's a really great application for this method and analysis approach. And then finally, using DIA might help us discover new PTMs, because it's an unbiased sampling, especially of those isobaric post-translational modifications. So thank my lab and especially my funding. And yeah, I think future applications for DIA, if anybody wants to get working on things like protein-protein interactions or protein localization, I think these are great places for DIA to expand in the future. So go ahead, and I'd be happy to take any questions that you've got, or even start a bit of a discussion on some of these things.
Lindsay, I thank you very much. It's great to see all the possibilities. I know you've already helped me with some DIA and SILAC data that we're just getting started to look at. I'm really excited about that possibility. I wanted to tell the attendees that when you showed your different schemes, we've been using 8MZ windows, and that seems to work well. We've done some evaluation, but you could spend months trying every different combo, and in the end, find something that works well. And the 8MZ windows for us has been just fine.
Yeah.
And with
What
the
instrument
3D
is that
space,
on? The 8MZ?
pardon?
What instrument is
The
that on?
Lumos. It's on the Lumos.
Yeah.
Part of it is because they're self-pack columns. I'm the one who packs the columns, and it's the same packing material I've had since the late 1990s.
Hey,
they're probably
it
not.
works.
Yeah, I'm not changing. They're not packed as tight, perhaps, as some people. So my peaks aren't ultra sharp, but seems to give us excellent results. And the thing that I did want to also mention that I hadn't said is we're now exclusively using ProCit libraries for all of our analysis, but also in conjunction almost all the time with a chromatogram library.
Oh, yeah.
On a few cases, we will acquire samples with 2 gas phase fractions across for each sample. So it's 2 injections of each sample, and then search a ProCit. But that's not for routine. Regularly, we would always make a chromatogram library. But the ProCit has just been phenomenal because we've got all kinds of different species. We ran some marmoset samples last week. It worked just fine. As long as there's a FASTA database, then you can generate or have generated for you a ProCit. And it's just amazing.
Yeah, and we're happy to generate ProCit libraries for anyone that needs them. It's a pretty simple process, but if you send us a FASTA and some parameters, we're happy to do it. Yeah, and all you need is a FASTA. So as long as there's a good one for the species you're working with, it doesn't have to be human. It doesn't have to be mouse. It doesn't have to be Arabidopsis. I've made some pretty obscure ones for Sue. So
Yeah,
just as if you're not studying a model organism and you still want to take advantage of the benefits of ProCit, we can definitely make that happen. So it's been a really good service, I think, for
yes.
both for us
yes.
and for people in the community. I think the results look awesome for everybody that I've at least gotten feedback about. So people seem to really like it.
I couldn't manage really without doing it. But what's great is you acquire the data and you can keep reprocessing it. So if you have only a so-so library right now, I mean the FASTA right now, then as something gets better or better annotated, you just can research it.
Yeah, totally. And they're reusable, as long as you're working with that organism, you can keep using it. So it seems to, the results look good, and it seems to cut down on some of the processing overhead, which is what you want, right? You don't want it to be easy and not give you good results, but it seems like it's doing both. So,
and you can combine 2 DLibs that you have, 2 ProCit libraries. You can combine them in Scaffold because you can only use one library at a time. But if you have, for example, 2 species that are mixed together, then just combine the libraries and do the processing.
True. Excellent. Okay, well, does anybody have any final comments?
Yeah, we did have some questions, some Q&A in the chat.
I will. I'm not the person that should be answering those. So if anybody wants to, wants to take a stab at any of the, I can answer proteome software ones, but not the real scientific ones. So if anybody wants to look at those and take a stab any on the panel, feel free to do that.
Yeah.
Or if they're directed at somebody specific.
There's a question for Dr. Weintraub. How different are the data analyzed with ProCit libraries versus those analyzed with chromatogram libraries?
Okay, so let me be clear that we're using both of them because while it's technically feasible to search your DIA files directly against the ProCit library, you want to generate your chromatogram library against the ProCit, and then you search your experimental results against the chromatogram library. Before we had the ProCit, the best results were using some kind of a spectral library. Now, I will say that people who are working on one cell type or one species and have the time to generate a really deep DDA library, you're going to get the very best results that you can. But that's a huge number of injections and analyses. And most of us, and especially in a core environment, we can't do that. So again, the steps are you get the ProCit library, you acquire your gas phase fractions, generate what we call the ELIB, the chromatogram library from that search, and then use that ELIB as the reference for your experimental samples. And the way I usually do it is the gas phase fractions will run one block first, then acquire the gas phase fractions, and then start acquiring the rest. This is all programmed, of course, in the instrument. And while this is going on, I will generate the chromatogram library while the rest are being acquired and search the first set just to see how everything is. And plus, because I can't wait to see what some of the results are, and then process the rest.
Yeah, and so you bring up a good point that it's beneficial for everybody, but it seems like you in a core facility type environment have been able to take advantage of them tremendously, because it allows you to not have to build an individual library for every new, because you're not getting the same thing every time, right? So if you need to jump between species, in between samples, it allows you to do that a lot more easily than have to build a chromatogram library every single time for some, maybe for the the smaller type experiments that it's just not feasible to do so.
Even some related cells, I've been amazed, investigators have sent the cells, and when you look at them in DIA, there's clear differences between them or among the different groups. And so it just is not feasible in a core laboratory to acquire that many.
I think as well the fact that the library is made with the client samples, and there's no sort of intellectual property conversation regarding the source of the library, what's going into the library, and how the library is made. Eliminates a huge piece of work. You don't have to have the conversation about intellectual property related to the library. And two, like Sue said, you don't have to make a good library. It's going to take a lot of time, and it's going to have a cost overhead associated with it, which is probably going to prevent you from running samples that you want to put in your study. And so when you look at this, when you take a step back and you eliminate the overhead with generating the library, the DDA library, and you look at the decreased runtime associated with getting the data, the net is just you can increase the scale of your experiment and start to pull in all the controls you want to do without running over budget. It's a really cool piece of software.
Yeah.
Definitely.
Lindsay, do you have any comments on the question about FAMES?
I actually, this might be a good one for Jacob. I don't know, is ion mobility, like FAMES ion mobility, built into Scaffold yet?
It's not right now.
Yeah, on the list, I'm sure.
Yeah, I think it's pretty high on the list. At least, it's something that we've certainly talked about, but it's not something that's been implemented yet. But if that seems where, if we see that people are interested in that, then it's something that we could definitely consider adding in the future.
I think there is a workaround. I think if you use MS Convert, I think there's a way in MS Convert that you can parse out the different CV acquired spectra into separate files and search each of the. I think there's a way to do that, but I have not done it
I've been
myself.
exploring this to see if I want to put FAMES on a shared instrumentation. Granted, I've going to be resubmitting, unfortunately, and it's something I'm going to have some discussions with people who have been trying it. But one possibility is, while you can't take advantage of all of the features of FAMES, and I'm saying FAMES specifically as it's on a thermo instrument rather than ion mobility in general, but you can help eliminate the 1+ ions going in. So you could set it at a single setting, and it might let you dig down deeper without having the 1+ even there. When I've used different settings for the charge states, I used to use 1 to 4, and I now really stick with 2 to 3 because your best data are going to come from 2 to 3, the charge states. So I'm hoping I can get somebody to give it a try so that I can comment on it on a shared instrumentation grant.
Yeah, and that's one of the things about when you generate a Proteome library, it lets you choose what shard states you want to use. So we've gone back and forth between making some with 1 to 4 and 2 to 3, and it's pretty trivial to do it from 1 to 4 and then try it from 2 to 3 and see how the results look and everything. So so it provides a lot of flexibility. Yeah.
And as I said before, I'm really happy to meet separately with anyone who would like to look at the data in depth or have some discussions of ways that you can use DIA or how you can interpret the results.
Okay, well, with that, I think we've gotten through all the questions, so I think we'll go ahead and end our second virtual brunch. Extra special thanks to Michael and Sue and Lindsay for joining us, for talking about some science. We had we talked about DIA, but a lot of stuff in the DIA space. We talked about, you know, analyzing some data, some new stuff that's coming on. All the presentations were awesome. So again, thank you very much. If you have anybody has any questions, feel free to proteomesoftware.com. You can submit a support request or send us an email. Mike, what's a good way to get up with you if you for people that want to reach out to you? On your website, or
you can go straight to info@MS Bioworks. A quick acknowledgement to you and the folks at Proteome Software, because, you know, with the pandemic, everybody's looking for wins, you know, and we're seeing, like Sue said, people have been working through this, you know, and it's not been an easy time, and you've delivered this product. It's amazing. It's a real highlight. I think it's a fantastic job.
Well, great. Well, thank you very much. Yeah, it's been nice to you know, at least get together, at least on Zoom, and talk about a little bit of science. And who knows, maybe ASMS in October will actually happen this year. So
there you go.
In a couple months, we'll be seeing people face to face, which would be nice. But this has worked out pretty well in the interim. So with that, I'll go ahead and say thank you, everybody. Thank you, the attendees, of course. Plenty of ways to get up with us if you have any questions. And yeah, thanks for joining us. We'll see everybody soon.
Take care.
Bye.
Cheers.
Bye.
Bye.